Go from the beginning. Here we are. Okay, that's just me. We've already done this. So what we're going to do today is I'm going to talk briefly 3 different times, 3 different kinds of topics, starting with aerosol transmission, then a little bit about the epidemiology of person-to-person transmission in clusters, and then we'll talk about control banding. And as you know, there are going to be 3 breakout groups. People have already been designated as discussion leaders and note takers. For a couple of the breakouts, I will invite, randomly invite people to report. We can't obviously do all 19 groups. The last one, I probably will just review the answers because we probably won't have a lot of time for discussion. We'll have a very, very short time for a break before the 3rd breakout. And we would like, if you could, the note takers to submit the completed exercise forms or your notes or whatever works for you. You can just send those back to Diane, and do bear with me because this is a pretty new learning and teaching format for me. So what I want to emphasize is that at this point in a pandemic, and we're still pretty early on, we actually really don't know how this organism, SARS-CoV-2, is transmitted. What we do know is that it's possible there are a number of different routes of transmission. And I've listed the top 3 are the ones that are traditional for infection control people in hospitals, where they talk about contact, which means transferring something from a source or an object on your hands to your mucous membranes, your eyes, your nose, or your mouth. Droplet, which is considered large droplets, large spray droplets that are propelled into your face and onto your mucous membranes. Generally and traditionally, that's not considered inhalation, and it is usually only thought to occur fairly close to an infectious source. Airborne is a word that's used for very rarely and only for a few organisms and diseases by infection control, and it has to do with these larger spray particles evaporate to what's called droplet nuclei. They travel far away, and then someone inhales them. And so that requires, that is inhalation, but it usually requires that it occurs quite far from the source, and by far, I mean even outside of a room. So measles and TB, for example, tuberculosis, are usually referred to as airborne diseases. But, and it's not thought that SARS-CoV-2 is airborne, although I would argue that we actually don't know if that's the case. And something I'm going to talk about, which is new to this paradigm, this infection control paradigm, is aerosols. And the way I define aerosols is that they are smaller particles inhaled near the source, so near the person who's generating them. I'm going to talk a little bit about how they get generated. But I do have to admit that the word aerosol, is the way I use it, doesn't just mean small particles. It actually is a very broad term that means simply a suspension of particles in air. And I've put a little picture here of a spray aerosol. Basically, a spray aerosol is what we're mostly talking about when we talk about human-generated aerosols. They're liquid droplets. They're and some are large and some are small. And you know, what you generate out of an aerosol can, for example, is a good example of a spray aerosol. There's a lot of liquid that evaporates fairly quickly, especially if it's water, as in the case in human-generated aerosols. So you'll have a quick change in particle size distribution from larger particles to more smaller ones. And you're going to see some figures here in my presentation that I wanted to be sure to sort of set the stage for. So when aerosol scientists talk about particle size distribution, we are talking about the range of sizes that particles occur in. And if you look at the left graph here, what you see is what, if I were to put that distribution on a linear scale, and the linear scale is shown at the bottom, you would get what's called a log normal distribution, a long tail to the right. So you have a lot of particles that are pretty small, but you also have a number that go quite a smaller number, but they go quite far into to the right side, meaning quite large. And you can see this distribution says they go up to even 100 and maybe more. On the other hand, if I take those same data, that same information about size distribution, and I put it on a log scale, that's the thing on the graph on the right-hand side, then it makes it normal because it's log normal. So this is the way you're going to see graphing occur, is this in the photos I show you, but you have to remember this is on the log scale. So what it means is there are a lot of particles in this larger size. But this normalizes so that you can see the median size, the geometric mean we call it, for this distribution is around 10. Well, you can see that that's probably the case here, but it's much easier to see here. So that's why partly why we do log normal distributions and on log scale, is to show you what the distribution is. Just to keep you give you a little, if you haven't heard of micrometers before, which I assume most of you probably have, they're pretty small. They're about a millionth of a meter, and a meter is about a yardstick. A human hair is 80 to 100 micrometers, and we can see things as small as 50 to 60. Maybe some people even can see a little smaller than that. So those are pretty large droplets in the way I think of aerosols, is the range of our ability to see. When we think about human-generated aerosols, in the last 10 to 15 years, there's actually been a great amount of research on this. How much do people actually generate? And for breathing, what we found is that, and again, this is on the log scale, so I want to make sure that you are aware of that. And you should just look at this sort of solid line. This is from work by Lydia Maraska's group in Australia. And what she's showing is that you get a mode, the geometric mean is about 1 or a little less than 1. But when you breathe, you're breathing out particles that are quite small, about 0.1, all the way up to about 10 microns. That size I think of as pretty small in the context of, let's talk about speaking or voluntary coughing, for example. And again, just look at these solid lines, and what you see is what we call a bimodal distribution. There's a bunch of small particles, and they're similar in the size of breathing, that they seem to hover, have the maximum around 1 or maybe 2 microns, but they also have this big chunk of larger particles that happen up around 100 to 200 micrometers. So you've got a whole ton of large particles, and large particles could, they will be able to carry more viral organisms, each of them, than these smaller ones, but you have many more smaller ones than you do large ones. So basically we get mode 2 peaks at about 40 to 140 for sneeze aerosols. So these are the largest of all of the aerosols that we generate. And sneeze is a high velocity, coughing is high velocity, sneeze is very high velocity. So quite a lot of stuff, but you'll see the same sort of bimodal distribution, but it's pushed over to the right a little bit. Here's another example of coughing from healthy subjects, and the reason I'm showing this is as they start out at droplets, they actually occur in maximum number at 1 and 2 and 8 microns, but as they evaporate and they form what's called droplet nuclei, they start to mostly hover around 1 micron, although you do have, of course, a wide range in terms of distribution. But many of them are about 1, and 1 is pretty small. 1 is something that you can breathe into and it will deposit in your lungs. And that's what I'm going to talk about in just a minute. But one thing I want to say is people are very variable in the number of particles they generate. This shows 8 or 9 subjects, and it shows them when they were infected with influenza, that's the dark bars, and then after they recovered. And while number 5 looks like they're not too different in terms of the numbers of particles per cough, there's a lot of difference for the rest of these people where when they were sick, they generated many more particles when they were coughing. But what's interesting also is the variability from person to person, where number 5 generates lots more particles than everyone else. This is what sometimes people call super spreaders. but I usually discourage the term because it somehow labels people as if they're sort of different, or I would suggest that this is the same kind of variability you see in people in terms of height or weight or all sorts of other biological characteristics. So this is simply the nature of biology. We are highly variable in many aspects. People generate aerosols between this 0.1 and greater than 100, as I just showed. So I want you to look at just this part of the graph, and it is a little confusing, and I tried to find something a little less busy, but what I hope the takeaway message here is what this is showing is particles in terms of their size, where they're deposited in different parts of the lung. So in the extrathoracic region, or what I would call the upper respiratory system, your nose, up down to about your trachea or your pharynx, those are where the larger particles get collected. And in many ways, our respiratory system serves as a filter. It's filtering out all that large stuff we breathe in, so it doesn't, the large things don't make their way all the way to the smaller, the air exchange region, which is the really important part of our lungs. And you basically want them to work well and not be blocked up with particles. So this is a filter, but you can then have smaller particles that somehow don't get collected in the upper respiratory system, making their way into the tracheobronchial region. And then there's a small, all these little alveoli, which basically are where the gas exchange occurs. And as you can see, you will get some deposition of particles around 1 to 10, but the amount of deposition, this is, you could think of this as percent, this is 20%, 40%. So you can see only a small fraction of the particles you breathe in that are in this range will actually deposit in our lungs. And so the key is big stuff here, medium stuff here, and little stuff here, but most will be here, a moderate amount here, and a very small amount here in comparison. But this is the key to understanding what happens with human-generated aerosols that are infectious, is that those smaller ones do have the possibility of making it all the way into the lungs and into the gas exchange region, and this is where many of the receptors are for SARS-CoV-2. So when we think about that classic disease transmission paradigm, this is an illustration of it. You basically they think of it as a source being coughing or sneezing, not talking or breathing. So talking and breathing and understanding that those generate aerosols is a pretty new piece of the puzzle in terms of human-generated aerosols. And many people in the infection control world do not really understand that we generate quite a lot of particles. They think of it as sources as mostly coughing or sneezing, but since there's so much asymptomatic transmission going on, meaning without symptoms, meaning people are not coughing or sneezing, we need to think beyond this droplet transmission and airborne transmission to something quite different, which I'm going to illustrate in a minute. But what I want to demonstrate to you is that when a person generates an aerosol, and it could be coughing or sneezing, but it can also be breathing or talking, they generate this wide distribution, and our artist did a nice job of showing by different colors and sizes the range of particle size that you generate. It isn't just all large particles that drop to the ground, as is often described by infection control, but rather it's an aerosol wide size distribution. Yes, the larger ones will evaporate quickly, and many of them will fall to the ground, and I'll show you that in the next one. So as time goes on, you do begin to get deposition of these larger particles onto the floor and onto the surfaces. The medium-sized particles are beginning to distribute, and the larger ones are distributing as well. So in the first picture, that's what is often referred to as droplet, propulsion. So if you're standing near somebody who coughs or sneezes, you can get things in your mouth or your nose and eyes. But once this happens, these are suspended particles that can be inhaled. And after some time, those particles, the smaller ones in particular, get distributed throughout the room and can also be inhaled by someone further away. And so, and people often ask me, what's the distance here? The distance isn't something you can necessarily put as a rule, because it depends on the size of the room, the ventilation in the room. And I'm showing you in terms of still air, as if there's no ventilation. The ventilation would, airflow would pick up these particles and move them further throughout the room. And if it's good ventilation, hopefully remove the particles from the room. But you can see you have inhalation could continue at both the close person and the far person. And I'm just briefly going to show you that these particles, I outlined the ones we probably care the most about, which are the 3 and the smaller, the 0.3, that they can remain in still air, suspended for quite a long time. And adding ventilation doesn't necessarily mean that they're all driven to the wall or to the floor, but that they can simply be transported even further, as I said, maybe out of the room if it's good ventilation. And so I've suggested that we add aerosol transmission, which I define as inhalation of small particles at close range, to the droplet and the airborne transmission paradigm. And I, for many years, tried to get people to change the definitions of airborne and droplet, and I gave up and just added aerosol transmission. Although really, particles, airborne is aerosol transmission, just further along, further away. And all of this aerosol transmission is related to particles, whether they're propelled into your face or whether they're inhaled. It's all related to aerosols. But so I didn't want to confuse you, but I do want to be clear that when I talk about aerosol transmission, I am particularly talking about inhalation of small particles, particularly at close range, but also can occur further away.